Welcome, and thank you for joining the webinar today. I am Clint Rose, Chief Scientific Officer and co-founder of Purigen Biosystems. Today I'll be talking about the system we've developed for extremely efficient nucleic acid extraction, the Ionic Purification System. A brief background on Purigen Biosystems, the company. Our mission is to develop a revolutionary sample prep solutions to help customers like you get the most out of each sample. Our core technology was invented by Professor Juan Santiago and his team at Stanford University. Professor Santiago is a co-founder of the company as well. The company is located in Pleasanton, California. That's in the San Francisco Bay Area. And we're neighbors with many other companies you probably recognize. We are backed by both Agilent and the Roche Venture Fund. So let's talk about the techniques for nucleic acid extraction that most users are familiar with, and some of the problems they're likely to experience. So most researchers are utilizing methods that are more than 25 years old, be they columns or beads. And frankly, the technology really hasn't kept up with all of the developments with the downstream analysis. What problems does this lead to? Well, you can get relatively low yields. You can have a lack of purity. You can get damage to the nucleic acid. One thing you definitely see is lower efficiency with small samples. And many of these methods are manual, meaning you could have errors, or they can just be tedious in terms of the workflows themselves. What's the consequences of that? Well, whenever you're thinking about the analysis, you should think of all that nucleic acid as being information that you want to go into either your research or into the clinic. And for researchers, a loss of yield or lack of purity can lead to reduced biological insight, just learning less from each of those samples that you have. For clinicians, this can mean less actionable information and the potential for false negatives. So let's dive a little bit deeper into traditional sample prep versus the method we're using called isotachophoresis, or ITP for short. So in traditional purification, whether that's column-based or bead-based, we're taking advantage of solubility. So we're utilizing chemistry to actually bind nucleic acid onto the surface. We're then washing across that surface, changing the chemistry one more time to then elute this material back off of that surface. Now, because this is a surface-based technique, what that means is you have the risk of loss and potential for damage. Loss, because material may never bind to the bead or column to begin with, or it may not come back off of that bead or column. And damage, because as we're trying to remove that material back off that surface, you can think of it as sort of tearing off of that surface, potential to break up the material itself. Other risks, potential solution contamination as we're doing these wash-throughs, and the risk of bias. And I'll talk about this a little bit later for things like GC content. Now, contrast that against what we do with ITP. In ITP, we're actually taking advantage of the inherent charge of the nucleic acid. Nucleic acid is very highly negatively charged. We're going to utilize this with an electric field to actually perform our extraction. Because of this, we're solution-based, which means we can get very high yields, very high integrity for the material itself, very high purity. ITP is actually inherently a separation technique as well, so we can separate away from the impurities within the system. The benefit of this is that we can give you the best representation of the sample. And what I mean by that is we can deliver as much of that starting material as possible. We do so without creating any bias or damage to the nucleic acid. And the end result is really what you care about most, which is better data. So let me describe in a little bit more detail isotachophoresis and how it works. So isotachophoresis is really based primarily on two buffers. The first that I show in the upper right with the blue dots, or leading ions. These are just fast ions that we've designed for the system to be much faster than the nucleic acid. There's a second ion with a second buffer that we call the trailing ion. And this is an ion that we select so that it is slower than the nucleic acid, but faster than everything else in the system. With these two buffers, we also have a third very important aspect, which is that we've developed our own lysis buffers. And these lysis buffers have been designed so that we can get access to all this nucleic acid, removing anything we need to, like crosslinks, if you're using something like an FFPE sample. But the end result is free nucleic acid in a solution that is immediately compatible with the ITP process. Once we have these buffers, we then utilize the fluidics in our chip to actually line these up in series. So in the back, you have your trailing ions, the red ions, in what we call our cathodic buffer. Next, we have the sample that contains our nucleic acid or your lysate that you've prepared. Next to that, we have our separation buffer that's containing all these leading ions. And then a fourth very important buffer is our extraction buffer. And you can think of the extraction buffer as being another version of this separation buffer, but at much lower ionic concentrations, so it's immediately compatible with all your downstream applications. Now, once we have all these lined up in series on our chip, now we can turn on the electric field. When we turn on the electric field, all these ions will start to migrate within the field through the channel, and all of your blue fast ions are going to race to the very front. Next, you'll have your nucleic acid. Nucleic acid is not fast enough to go ahead of the blue ions, but it's faster than all these red ions. And so it will speed ahead of the red ions, and it will sit at the interface between the blue and the red. Because nucleic acid is so fast, has so highly charged, it is faster than most of the impurities within the system, and so those impurities are actually dropping back behind, so you're getting a separation effect at the exact same time. Now this process continues. This band is physically moving through the chip. It will continue until the nucleic acid arrives in our extraction well. At that point, the system detects its arrival, shuts off the electric field. At that point, you can come in and extract that material. So now that you understand the principles of how ITP works, I'll explain to you the system that we've developed to utilize this for your nucleic acid extractions. What you see in the center is our ionic purification system. This is designed to be a benchtop instrument. It's about the width of a laptop, so intentionally trying to take up as little space as possible on your benchtop in your lab. It enables automated purification. It's touchscreen driven, so very simple, easy to use. And for each application that we develop, we have a application-specific chip. That's what you see here on the left, as well as a special set of chemistry that's really optimized for that given application. For each new application we develop, we simply develop a new chip and a new chemistry to go with it that works with the existing instrument. The output of that is a validated set of kits and protocols, each giving you the very high yields, high purities, and simplified workflows for each of the sample types you're working with. Now, let me take everything I just described to you and show you what this actually looks like on the instrument itself. So here you see the instrument. We'll open up the front of the instrument. You can see where the chip goes in. What I'm going to show you is an example of what this looks like as we're actually running the ITP process. So here I'm showing you a typical chip for us. You have 8 lysates that are going to run in parallel. In this case, I'm showing you genomic DNA that we've loaded into this sample channel. These are the stripes that you see. And we've added, just for demonstration purposes here only, an intercalator so you can actually see the genomic DNA moving through the channel as we run the ITP process. So as we turn this on, the electric field is turned on. We've sped up the video for this demonstration, but you can see the material collects all of the DNA. We move into the band. We continue to move to the left. This is where we're getting that separation effect. All those bands move into their elution well. The system detects their arrival and stops. This is showing you, again, the same process, but this time showing you with the ions as I described to you before. As you can see, the TE ions in the back, the LE ions in the front, we're getting this concentration effect of all that nucleic acid moving to the interface between the two of them, and the separation effect as those impurities are dropping back behind. As I described before, as the material continues to move through the chip, you're getting more concentration, more separation, finally arrives in the elution well. At this point, we come in with simple multi-channel pipette, and pipette out that purified nucleic acid. So let me describe the workflow to you in terms of how we actually load the chips, getting them prepared to run on the instrument. So this upfront workflow, very simple. We're going to utilize a multi-channel pipette, load our 5 working buffers into the chip. That's 5 simple pipette steps. Once the buffers are loaded into the wells on the chip, the instrument will close, performs a 2-minute priming step, and that's just putting all those buffers into the correct order within the chip, getting them ready to process the sample. Instrument then opens after those 2 minutes. User loads the 8 lysates to go into the system, presses go, and at that point, In about 50 minutes, all 8 of those samples will be processed. Instrument will let you know it's done. Come back, open up the lid. Simple pipetting step to remove all that extracted DNA from your extraction wells, as you can see here. Now we have 2 kits currently available for the Ionic system. The first kit is our cells to pure DNA low input kit. The advantage of this, we provide 50% higher yields on average from cells, and this is all the way down to 10 cells up to 100,000 cells of input. Second kit is our FFPE to pure DNA kit. And for this, for each of your sections, we provide 3.5 times more yield on average from those sections. So I'm going to give you a little bit of data for each of those 2 kits. I'll start with the cells to pure DNA low input kit. Now, the advantages we provide for extracting from cells, first of all, is we get 50% more DNA from the cells. We're doing so without using carrier RNA. However, that comparison is against standard column kits that are using that carrier RNA. So 50% more material. What you see is very good linearity all the way down to 10 cells up to 100,000 cells. And I can tell you we're recovering about 80 to 85% of the material that started in that lysate. I'll also mention that currently that kit is limited at 100,000 cells. However, we're working right now in development to further extend that upward into the 1,000,000 cells and beyond. Now, the other advantage I talked about previously is just talking about the quality of that DNA that you're able to get. So this is a very simple comparison. We're looking at the fragmentation of the material, whether it has gone through the Ionic system or an automated column-based system. So in this case, we're extracting genomic DNA from about 50,000 cells, and these are just replicates from that same starting cell stock. What you can see is across these 6 replicates, in each case for the Ionic system, you have this primary band of very long material, right around the 40K mark, with very little shearing of material in between, meaning we're not damaging the material within the process itself. When you look at the automated column-based data, the same type of 6 replicates, you can see one primary band, but a fair bit of shearing, meaning the material is actually being damaged in the process itself. I'm showing you this data in a similar way, in this case against manual kits, manual column-based kits. And now I'm showing you the percentage of material that's greater than a particular length versus the lengths that we were measured. And really what you care about here is where's the 50% point? Where is half of my material greater than or less than a certain length? And what you can see for the Ionic system is when we extract from cells, we're at about the 40 kb mark. Half of our material is actually greater than 40 kb. You can see for the column-based kits, it's significantly shorter. It's generally about 16 kb for the columns. Now I'll transition and talk to you about our second kit, the FFPE to pure DNA kit. Let's talk briefly about FFPE. In general, we all know it's wonderful because it is an incredible time machine of information. I've had great discussions with customers about samples that they want to extract from that may be 10 years old, 20 years old, in some cases 50 years or more older. The problem, though, is that that material, just inherent to the fixation process itself, is often very low quality. It can be very difficult to get good yields out of those blocks. And obviously, some of the workflows can be quite horrible. So we've tried to address all 3 of those issues, and starting with the workflow. This was very important to us in our development. We wanted to go from something like you see on the top with the column-based kits, many steps, starting from removing the paraffin, all the decross-linking, lots of washes and spins to get to your final DNA. We've tried to simplify that process down to simply taking your scroll or slide, dropping the material into a tube, adding our lysis buffer, putting that on a thermal mixer with an automated protocol that allows you to do the incubation. This process removes all the crosslinks. Once that's complete, now you have your lysates, you're ready to go into the purification in the process that I described to you previously. Once the system finishes its operation, as I said, about 50 minutes from the start, now you have your ready to use DNA. I have some data that we provide based on the performance assessment where we looked at a 32 block study looking at the Ionic system versus column-based FFPE kit. For this 32 block study, we have 3 different tissues, breast, colon, and lung. with blocks that are ranging in age from 2 to 26 years old. And the comparisons we're providing are across 4 different things. First is amplifiable yield, looking at DNA quality, as well as Amplicon sequencing data and hybrid capture-based sequencing data. Let's first talk about an amplifiable yield comparison. So for amplifiable yield, we're using QIAGEN's Q biomarker MRAP assay. This is a great assay when you're trying to look at small amounts of material. It has an 80 base pair amplicon, and the information it's providing you is how much amplifiable, or as we think of it, usable DNA do I have in each of my eluates. Now, here is the data on amplifiable yield from that 32 block study as I described, and I'll take a minute just to orient you to this data. So across the bottom are all 32 blocks. We're using simple identifiers for each of the blocks. In each case, we're using multiple sections from the block to see how repeatable the data is, and these are interdigitated. So one block's going into the ionic system, one block's going into the column-based. You can see that we've segregated into the 3 different tissue types, breast, colon, and lung. And the data I'm showing you is actually the concentration of the eluates for each of the individual slices that we put in. Although it is concentration, I will point out that we are eluting in approximately the same volume in all cases, about 40 microliters, which means this also translates into a total mass as well. And what you can see, Purigen in blue, column-based in red, is that across nearly all of these blocks, we're getting significantly higher amplifiable yields using the ionic system than what we were able to achieve with the column-based system. So the next important factor here is what's the quality of this material? How usable is this material going into my downstream assay? We have an assay we like to use, the Agilent QC-Plex assay, that's great if you're doing an Amplicon-based sequencing assay next. It provides 3 important pieces of information. The first is it assesses the DNA quality, and it does this by looking at 6 different lengths of amplicon, ranging from 100 base pairs up to 700 base pairs, and it's really just looking to see at what different lengths you can actually amplify based on the same sequence. It also has a 150 base pair fragment, and that acts as a PCR control. That's just ensuring that there are no inhibitors within the elute. The output of this is a DQC value, which is just an indicator of the quality of the material itself. And that might range from excellent to low. The recommendation then is how much of this material should I use, ranging from 1 microliter to not recommended to continue into your sequencing assay. So using this assay and taking the exact same 32 blocks that I just showed you, we're showing you the DQC score. This is a log scale on the left-hand side, and you can see the 4 different ranges, as I described previously, going from excellent to low quality. As before, all of the blocks are shown for you here with their block identifiers along the bottom. Blue bars for Purigen, the red for the column-based. And one thing I wanted to point out is that in 14 of these samples, in the column-based case, we actually resulted in no detection of amplifiable materials. So these are the dots down here. And what you can see is across nearly every single block, Purigen is providing higher quality material out of the exact same blocks than what we're able to achieve with the column-based system. So next, let's talk about what this actually means as you're going into your NGS analysis. Now, in order to provide a comparison on the NGS data, one of the important goals here is to make sure we normalize the FFPE sequencing data against a reference. The problem is we can't go head-to-head showing you 2 different kits because then you're making an inherent assumption that one of them is correct. Instead, what we're going to do is go against a reference that will remove all of the potential biases and other things that may just be inherent in those processes. So our approach to do this is we're going to generate a coverage profile of the assay using a high-quality reference sample, in this case, Coriell DNA. What that does is it accounts for the amplification efficiency differences that might be due to fragment length, GC content, or anything else. Once we have this coverage profile, we know what sort of the right answer is. We're just going to compare the Purigen sequencing results and the column kit sequencing results against that coverage profile to make our assessments. I'll walk you through that in 3 steps. So step 1, determine what the reference coverage profile looks like for the assay. So for one of our assays, this is what it looks like. These are the 252 targets within that particular assay. And when we sequence the Coriell DNA, what we can see is consistently the number of target hits we should be getting for all of those targets. Now, once we have this reference coverage profile, step 2 is we can start comparing the Purigen eluates and the column kit eluates against this. So we're going to take the number of actual target hits we got divided by the expected number of target hits. So if it's exactly the same, we should get a 1. We're then going to take the log of that number. And by taking the log of that number, we just create a 0 baseline. So 0 means we're doing exactly as expected based on the way the assay was designed. Anything above the line means we're getting more hits on a particular target. Anything below the line means we're getting less hits on the target. Then the third step is we want to compare this normalized data. So I'm going to show you this data broken out in a couple different ways. The first will be on a chromosome level. And the interesting thing about the chromosome level is just kind of helps us spread out the data so we can see some interesting trends. The second will be comparing this coverage data against certain parameters. Those parameters will be either length or GC. And you'll be able to see the performance against that particular parameter. Let's talk first about the Amplicon sequencing. So the assay we're using for this is Agilent's SureMaster tumor hotspot assay. As I mentioned before, this has 252 amplicons. Those amplicons range in size from 128 to 254 base pairs, and the size of the target region is about 26 kb. Now, this is the outcome of that data. This is for one of the blocks I showed you previously, block BM. I want to point out, this is a block which sort of average for us in performance against the column kit. It's neither our best block or our worst performing block. It's sort of in the middle. Now, when we look at this data, just to, again, orient you in terms of what we're looking at this log hits metric, as I described before. And as I said, the key thing here is you want to see the sequencing data tightly compacted around that 0 line. That means you're basically replicating what we would get out of high quality material, even though we're using the lower quality FFPE material. What you can see is generally for the ionic system, we're quite tightly compacted there. A couple of interesting things we see when we compare that to the column kit is that you get a couple of unique phenotypes in the data. One is this sort of island effect where most of the data on that particular, for the targets on that particular chromosome is shifted downward. The second is a tailing effect where you see all the data start to kind of tail downward on that particular chromosome. Now, when we dug a little bit deeper, as I said, we compare the coverage ratio against certain parameters. First, we looked at length, and this is the length of the target for that assay. For the ionic system, what you see is very good, consistent coverage independent of the length of the target. For the column kit, you can see as you start to get up to these longer targets, that's where these tails start to come from, and you start to get lower coverage. Now, the second parameter we looked at was the percent GC content. Again, for the ionic system, what you see is very consistent coverage, all the way from low GC content to high GC content. For the column kit, you can see what seems to be hints that there might be some bias there, but unconvincing. However, if I take the exact same data that I just showed you, I'm just going to highlight 2 of these areas. The first, I'm going to show you this little island. I'm going to highlight that in purple so you can really see it here. And when I highlight in purple and I plot those points in purple in the GC, what you can see is that that is actually GC bias, where the high GC material is actually getting lower coverage. The second thing I'll show you is that if I look at this tail in teal over here, as well as the 2 low performing points off the purple, you can see both of those are actually the long targets. So we are seeing some bias from the column kit that we're not seeing in the ionic system. The important question though is, does this matter? Does this actually translate into useful, actionable information for you as a researcher or you as a clinician? And the answer is yes. So the next thing that we did is we actually took blocks and we wanted to look at the detection of variance. Now, in order to run this experiment, what we did is we took the exact same block and we took either 1, 2, or 4 5-micron sections into the lysis. We did this for both the ionic system and the column-based system. We then ran it through the sequencing assay, and we looked at the coverage bias first. This is exactly the same type of data I just showed you, except it's all of the data for all of the 252 targets in one single plot. So you can see we get very nice, consistent coverage independent of whether we have 1, 2, or 4 slices in the ionic system. For the column-based system, you can see with a single slice, that seems to really struggle. You're starting to see quite a bit of tailing down, meaning loss of coverage at some of these targets. And even as we get up to 4 slices, in some cases, the coverage was still poor. How does this translate into variant calling? Well, we looked at the 26 variants that were in concordance across the 2 assays, and then looked at 1, 2, or 4 sections and said, how many of those variants can we detect based on the number of sections we put in? And you can see in the Purigen case, even with a single section in, we were able to detect 25 of the 26 compared to the column-based kit where we're only able to see 21 of the 26. And once we got to 2 sections, we were able to detect all 26 for the same thing. On the column kit case, you can see this coverage did matter, and it's showing up in terms of our ability to actually call those variants. So less coverage is translating into less variant calls, basically false negatives. We did similar analysis for hybrid capture-based sequencing as well. In this case, we're using Agilent's SureSelect Cancer all-in-one solid tumor assay. This has 98 genes that are targeted. The size of the target region is about 740 kb in this case. Now, the first step was we just wanted to get a sense of the high-level QC metrics from our sequencing. And to do this, we did a simple workflow normalization. We took the eluates from the exact same block, processed similarly to the way I've described before, where we're taking interdigitated sections off of the same block. We're taking those eluates, we're normalizing based on qubit before we go into shearing, so we know we have the same amount going in. We then normalize on the way out based on tape station, so we know we have the same amount of material going into the end repair and A-tailing. And then we continue through the standard process, PCR, hide capture, and into sequencing. And if we look at the high-level QC results from the sequencing, what we see is they're basically equivalent. The column kit is basically equivalent to the ionic kit in that the percent on target, the exact same amount, the percent duplicates nearly identical, the insert length nearly identical. The next layer, we wanted to look a little bit deeper, and this is a plot very similar to what I showed you before. This assay has much more information in it, and so I'm showing you the data instead of in the dots like I showed you. This is in a violin plot, but the same goal here. What we want to show you is spreading out the data by looking at it on a chromosome by chromosome level. What we want to see is we want to see very little spread away from this 0 point. You want all the data sitting on 0, very little spread away from it, and you can see that's what we're achieving with the ionic system. For the column kit, what you can see is you do start to get spread away from that 0 point. Question is, why? Well, when we look at this data and we look at the relative coverage against GC fraction, I think it becomes clear why you're starting to get that spread across all of those different chromosomes. What you can see for the ionic system is the coverage is very consistent from low GC fraction up to high GC fraction. For the column kit, as you can see in this case, what you get is some bias, some lower coverage at the low GC fraction, and then kind of normalizing back out as you get to the higher GC amount. So with that, what I'd like to talk to you about, now that you understand the existing applications we have, I'll talk to you about some of the future applications that we're currently working on. So I already mentioned the FFPE, the DNA from FFPE, DNA from low cells. We're currently working on and should have soon the RNA from FFPE and RNA from cells, as well as DNA and RNA from fresh frozen tissue, as well as some other applications we're working on ranging from blood and plasma to environmental and viral samples. Now, I'll show you a little bit of early data. This is pre-verification validation for us, but some exciting data that we wanted to share showing our FFPE to pure RNA. And what I'm showing you here is a comparison of the yield from the ionic system compared against a column-based kit. And the comparison I'm making is using the beta-actin ratio, meaning how much beta-actin do we detect versus that you can detect from the eluate from the column. And the red line that you see here is parity. We're looking across 15 different blocks, similar to what I showed you in the DNA case. In this case, the age of these blocks is ranging from 3 years to 24 years. This is our oldest block here. And what you can see across these 15 blocks is in the majority of the cases, we're getting more RNA out than what you're able to from the column-based kit. And actually in 9 of these cases, we got an average of twice as much material out. The second kit that we're working towards right now is the tissue to pure DNA. And this is an example of some of our early data as well. This is an extraction purification of DNA from rat spleen. As you may know, spleen can be a very difficult tissue to work with. And the data I'm showing you here is an example of the eluate concentration we're able to achieve using the ionic system versus the column kit. These are all tissue that is coming from the same initial tissue source. It's been weighed, so we're getting consistency in terms of how much actual tissue is going into each of these preparations, and these are individual extractions that you're seeing in each of these data points. So you can see from a concentration perspective, we're getting about 10 times higher concentration of DNA than what you're able to achieve with the column kit. So with that, just want to summarize, we're working with this ionic system, provides you much higher yields from your samples. The 2 examples I provided are the 50% more yields from cells, 3.5 times more yield from your FFPE samples. We've worked very hard to really simplify the workflows using the fully automated system that we have. And the output for you should be high quality NGS data, even if you're working with lower quality or limited samples. We definitely want to hear from you. We'd love to understand what sample types you're using, what workflows you have, and what challenges you're facing. So please reach out. If you have any questions or you're interested in the system, you can contact us either at either of these emails or directly via our website. Thank you.